And now we are going to see the details of the particular electron transport chain. So as I told you, there are 4 major complexes going to involve, and the 5th complex going to produce the particular ATP molecule. So the complex 1, 2, 3 and 4, and the complex 1 over here is NADH dehydrogenase. Okay, and the second complex over here, succinate dehydrogenase. So okay, as I told you in the Krebs cycle, so the succinate, the conversion of succinate to fumarate. Fumarate going to release the product FADH2, and the NADH is being released from the particular glycolysis and from the Krebs cycle. So as we all know, in each cycle there are 3 NADH molecules being released, and for a single glucose molecule there are 6 NADH molecules being released in the Krebs cycle. So over here it is also from the Krebs cycle, and both the molecules, both the NADH molecules and the FADH2 molecules are going to enter into the particular electron transport chain, and the hydrogens, the atom is being transferred to or converted into in the form of protons plus electrons, and the electrons is being gonna sequentially transferred to one carrier to the next carrier, and the proton is being pumped out of the particular area, and that will gonna create the particular proton gradient, and by means of proton gradient it is gonna create the energy to form the ATP molecule. Okay, and now we are gonna see the details how these things gonna happen in the electron transport chain. So this is a structure you can observe in the inner membrane. Just imagine this is a inner membrane of a mitochondria. Okay, and there must be some outer membrane. Okay, so as I told you in the previous section over here, in between the two, inner and outer membrane, there is a space called intermembrane space. Okay, so just forget about the outer membrane. Everything, all the process is going to happen in 3 regions. So matrix, what is matrix? The center semi-liquid portion of the particular mitochondria is called matrix, and the inner membrane, and the space in between the inner and outer membrane, that is intermembrane space. Okay, so to our convention we are going to call this intermembrane space as C side, and this side, the matrix side, as a M side. Okay, so this side is called M side and this side is called C side. C side refers to over here intermembrane space in between the two membranes. Okay, and as I told you, we are in the structure called inner membrane of the mitochondria. As I told you, all the electron transport process going to happen in the inner membrane of the mitochondria. Okay, so as I told you, these are all the protein complexes. It is being arranged in a particular sequence so that it can transfer the electrons from one carrier to the next carrier. So what do you mean by carrier? Okay, so over here you have to understand the term electron donor and electron acceptor or electron carrier. Okay, so normally the electrons will tries to move from the higher energy to the lower energy form. Okay, you should have studied in the chemistry. Okay, electrons will tries to move from higher energy to the lower energy. Okay, so the same thing going to happen over here. As I told you on the aerobic respiration, what is the final electron acceptor? Oxygen molecule. Okay, so as I told you, oxygen is a electron acceptor. Why the particular cell system going to use the oxygen as a electron acceptor? Because the electronegativity of oxygen is very high compared to the other relative atoms. So over here the electronegativity of atom, the particular oxygen atom will pull the electrons towards the particular atom. Okay, so over here the particular complex is being arranged in a sequence in that way. The each and every step, in each and every step or in each and every complex. Okay, so what is the final electron acceptor over here? Is oxygen. Okay, so the electronegativity of complex 4 will be less than the oxygen, and complex 3 will be less than the complex 4. complex 1 and 2, they are going to donate the electrons. Okay, that means the electrons from the complex 1 and 2, they are going to sequentially move down the particular energy level, okay, till they reach the particular oxygen molecule in the electron transport chain. This is what you have to understand about the electron transport chain. They are going to transport the electrons through the chain. Okay, so what is the ultimate principle over here? As I told you, from the donor to the carrier or to the electron acceptor through their electronegativity property. Okay, so the final electron acceptor over here is with the high electronegativity compared to the remaining complexes involved in the particular electron transport chain. Okay, and this is the first thing you have to remember about the electron transport chain. As I told you, everything going to happen in three regions of the mitochondria: intermembrane space, inner membrane, and the matrix. Okay, so as I told you in the previous section, the matrix, in the matrix all the Krebs cycle process going to happen. Okay, from the pyruvate, conversion of pyruvate into acetyl CoA, and the entering of acetyl CoA as a citric acid into the Krebs cycle, and all the 10 steps of Krebs cycle, and the synthesis of NADH molecule and FADH2 molecule, and all the remaining products again come back to the cycle, and the conversion of the pyruvate into the single carbon dioxide and other byproducts of the Krebs cycle. Okay, so everything going to happen in the matrix side. And the point you have to remember over here, from the matrix side, there are major products as I told you: NADH, there are protons, plus the FADH2 molecules. Everything gonna enter into the electron transport chain. This thing gonna happen in the inner membrane of the particular mitochondria. Okay, so as I told you, over here they are being arranged in a series of complex 1, 2, 3 and 4. As I told you, the one refers to Nadh dehydrogenase. Over here they are gonna accept the electrons and protons from the NADH molecule. Okay, so as I told you, they are gonna accept the electrons from the NADH molecule. So the process will be like this. Okay, so they are going to accept the protons along with electrons. Okay, and they are going to liberate the NAD plus molecule. Okay, so the point you have to remember over here, for each and every NADH molecule, it is going to produce approximately 3 ATPs per NADH molecule. Okay, this is what you have to remember. And about the next complex, 2 ATP molecules per FADH2 molecules. Okay, so this is what you have to remember to calculate the total amount of energy is being produced from the glycolysis or the complete carbohydrate metabolism, and we are going to discuss the details later. The point you have to remember over here, the first complex, they are going to accept the electrons and protons from the NADH molecule. Okay, and in the same way, the second complex, the succinate to fumarate conversion and the succinate dehydrogenase over here, they are going to liberate the FADH2 molecule. That will be accepted by the complex 2. Okay, and both the complex, they are going to transfer the electrons to the intermediary molecule. Okay, so just remember one thing, apart from these 4 or 5 complexes, there are 2 other carriers. Okay, we are calling those carriers as mobile carriers of electrons. Okay, so mobile electron carriers. So there are 2 mobile electron carriers also involved in the particular electron transport chain, and we are calling those carriers as coenzyme Q, or simply we can call that as Q. Okay, and cytochrome C. So we are calling these 2 as a mobile carrier. And the point you have to remember over here, the cytochrome C is a lipid soluble mobile carrier, and the cytochrome C is a water soluble mobile carrier. Okay, so just remember one thing, all the process of the particular electron transport chain gonna happen in a membrane. Okay, so what is membrane? We all know the structure of a membrane. So what may be the structure of any membrane? It will be like this. Yeah, hydrophilic and hydrophobic structures is being arranged in a particular order, and hydrophobic tails will be in the inner side and hydrophilic hydrophilic head and hydrophobic tails. Okay, so for any membrane the structure will be like this. Okay, it will be arranged in a sandwich manner. Okay, so the point you have to remember over here, all the process gonna happen in this structure. Okay, this is a single membrane. In a membrane, everything, the electron transport chain gonna happen in a membrane structure. Okay, so what happens over here, as I told you, the ultimate process, the transfer of electrons from one place to another place. Okay, so the electron should be transferred through the hydrophilic nature or through the hydrophobic nature. Okay, so they need carriers of both lipid soluble and water soluble, and that will be provided by the two structures. The coenzyme Q is a lipid soluble electron carrier and the cytochrome C is a water soluble mobile carrier. This is what you have to remember. Okay, so I will summarize over here from the complex 1 and 2. So complex 1 will receive the NADH electrons and protons. Complex 2 gonna receive the products of succinate. Okay, and over here they are gonna donate the electrons primarily to the intermediary mobile carrier over here, the coenzyme Q. Okay, so there is some other term for the Q ubiquitone. Okay, so as I told you, the entire process is oxidation and reduction, or we can call that as redox reactions. Some molecules gonna get oxidized and some molecule gonna get reduced. Okay, so over here from the quinone, okay, it is gonna donate or it gonna transfer the electrons to the next complex, that is complex 3. Okay, and the complex 3 gonna pump the particular protons out of the particular inner membrane through the intermembrane space. Okay, so this is what you have to remember. The protons released during the particular electron transfer will be pumped out of the particular inner membrane and they will be dumped in the C side of the particular mitochondria. C side means intermembrane space, that is the space in between the two membranes. Okay, and this is also made up of the cytochromes B and C1. Okay, and then from the complex 3 the electrons will be carried to or transferred to the next complex, complex 4. Here also the protons gonna pumped out of the particular inner membrane. Okay, and in the complex 4 the electrons will be finally accepted by oxygen, and to take up some hydrogen atoms they are going to synthesize the water molecule. Okay, and up to this the water molecule is being synthesized. The point you have to remember over here, the transfer of protons to the intermembrane space. Okay, so that is what we are going to discuss now, and that process, the synthesis of ATP process, is called oxidative phosphorylation. Oxidative phosphorylation. So why we are calling this as oxidative phosphorylation? As I told you in the previous section, you have to remember, in case of the photosynthesis they are also going to produce the energy by utilizing the sunlight energy. Okay, so that kind of energy synthesis is called photophosphorylation, because they are going to synthesize the energy by means of utilizing the sunlight energy or photon energy. Okay, so that process is called photophosphorylation. Over here the system gonna produce the energy by utilizing the oxidation reduction reactions, so that we are calling this production as oxidative phosphorylation. As I told you over here, okay, this is a mitochondrial inner membrane, this is a outer membrane, and by means of electron transport chain the protons from the matrix constantly pumped out of the particular matrix to the intermembrane space. Okay, so what will happen over here, that will create or that will concentrate the amount of protons in the intermembrane space. Okay, and the point you have to remember over here, the inner membrane is impervious to the particular protons compared to the particular matrix. Okay, and as we all know, the membrane is a semi-permeable nature. So it will not allow to transport the charged particle to the particular area without the help of proteins. Okay, so what will happen over here, it will create a gradient. kind of gradient they are going to create? They are going to create the pH gradient and current gradient. pH gradient. So what do you mean by pH? pH is a hydrogen ion concentration. In a solution, if you are going to increase the concentration of the proton, it will become a nature acidic. What is acidic? High concentration of protons is called acidic. What is basic? Lower concentration of protons are basic. Okay. So the same thing you have to apply over here. What happens over here in the intermembrane space? The higher amount of protons is being deposited. So that will create a proton gradient in and out of the particular membrane. Okay. So over here the protons will be very high and here very less amount of protons. Okay. So in the same way you have to imagine it will create a charge gradient also. It is a protons are positively charged ions. Okay. So the pumping of higher amount of protons into the particular intermembrane space will create a charge gradient. Over here the net positive and over here net negative charge, and these pH and charge gradient they are going to produce a environment called membrane potential. Okay. So as we have studied in the photosynthesis, the osmosis, the transport of molecule. Okay. So what is osmosis? The movement of the substance from its higher concentration to its lower concentration. Okay. So the same thing will try to happen over here. The protons will try to move from its higher concentration to its lower concentration. So over here in the intermembrane space they are in a higher concentration. Within the matrix they are in a very lower concentration. So the protons will try to enter into the particular inner side, that is the matrix of the particular mitochondria. Okay. So this is what you have to remember. Because of the membrane potential, the osmotic gradient also going to form, and because of this osmotic pressure the protons will try to enter into the particular matrix side. Okay. And for everything you have to remember one thing, the protons cannot pass through the membrane. Okay. They will try but they cannot pass through the particular membrane. They need help from some other proteins. Okay. And just remember all these complexes, complex 1, 2, 3 and 4 are proton pumps, but they will pump out the protons, but they won't allow the protons enter into the particular matrix. Okay. They are gonna pumping out of the particular proton out of the particular inner membrane. So there is only one possible way for the particular protons to re-enter into the particular matrix, that is through the complex 5. Okay. So through the complex 5 they are trying to enter into the particular system, and now we are going to see the details of the complex 5 and what is oxidative phosphorylation. As I told you, the complex 5 is otherwise called as ATP synthase or simply ATPase. So why we are calling this as ATPase or ATP synthase? Because they can perform two processes. Okay. From the ADP and inorganic phosphate they can synthesize ATP molecule. Okay. So while performing this function we are calling this enzyme as ATP synthase, because they are going to synthesize the particular ATP molecule from the inorganic phosphate and ADP molecule. At the same time, based on a need and based on the availability of electron donors, they can perform in a reverse direction. That means from the ATP they can create ADP and inorganic phosphate. That means they are going to break down the particular ATP molecule into ADP and inorganic phosphate. In that time we can call this particular enzyme as ATPase. Whenever you are seeing the term ase in an enzyme, they are going to break the particular molecule. This is what you have to remember. Nuclease means it is going to break down the nucleic acid. Carbohydrate, carbohydrates, lipase. Lipase means the enzyme going to break down the particular lipid molecules. So ATPase means they are going to break down the ATP molecules. The point you have to remember over here, at the same time they can create the ATP molecule, so that we can call this enzyme as ATP synthase, because from the inorganic phosphate and the ADP, they can create the ATP molecule based on the availability of the electron donors and the proton donors during the oxidative phosphorylation and the electron transport chain. Okay, so that is what we are going to see now. The complex 5. As I told you, in the complex 5, this is also present in a inner membrane. Okay, and in the inner membrane, there are two structures of ATP synthase. You have to remember F1 and F0. Okay, so there will be a protein stock and the F0 is being attached, F1 is being attached like this through the F0. Okay, so you have to remember one thing, the F0 is a proton channel. That means this channel gonna allow to enter the particular proton molecule, proton atoms into the particular matrix site. Okay, so as I told you over here, the protons is being concentrated over here. Okay, so they all try to enter through the particular complex 5 or ATP synthase. So in the ATP synthase, you have to remember there are two portions, F0 and F1. Okay, so the protons, all the protons will try to enter through the particular ATP synthase, and F0 is a proton channel, and while it enter into the F0, they are gonna rotate the particular F0 along with the particular F1 molecule, that is the F1 portion attached to the particular enzyme. Okay, so the F1 is a projection that is in a inner side, that is the matrix side of the particular mitochondria. Okay, so this movement, the movement of protons by means of membrane potential or proton gradient, when enter into the particular F0, they are going to create a proton motive force. Proton motive force. Okay, so by entering through the F0, they are going to create some force by means of particular movement. They are going to create the energy, and that energy will be utilized for the synthesis of ATP molecule, as I told you, from the inorganic phosphate and ADP. They are going to create ATP molecules. Okay, so the point you have to remember, as I told you, for each and every NADH molecule, okay, there will be 3 ATP molecules will be produced. For each and every FADH2 molecule, 2 ATP molecules will be produced. Okay, so as I told you in the glycolysis, okay, only you have synthesized 2 ATP molecules net production in the glycolysis, but we have produced NADH2 molecules. And as I told you, NADH2 molecules indirectly involved in the synthesis of ATP, and they are entering into the particular electron transport chain to synthesize the particular ATP molecules with the help of enzyme Atp synthase. Okay, so this is what you have to remember about the synthesis of ATP molecules. So what are the major things we have seen over here? Electron transport chain and oxidative phosphorylation. What is electron transport chain? With the help of the electron donors and the electron carriers, the continuous oxidation and the reduction sequence, redox reaction sequence, the electrons is being transferred from one carrier to the next carrier, and simultaneously the release of protons into the intermembrane space and the creation of the membrane potential. Everything going to happen in the electron transport chain. At the end of the electron transport chain, the oxygen will accept the electrons and create the product called water, and then by means of oxidative phosphorylation, the enzyme ATP synthase of the complex 5 going to synthesize the molecules called particular ATP. Okay, and this is what you have to remember. And there are 2 mobile carriers, lipid soluble Q and water soluble cytochrome C. This is what you have to remember. These are all the details you have to remember with the electron transport chain. And now we are going to discuss the amount of energy is being produced from each and every glucose molecule. Approximate amount of energy. Why I am using the term approximate? Because we cannot say that in each and every step. As I told you, so what and all we have seen till now in the respiration, we have started off with the glucose. Okay, enters into the cycle called glycolysis, synthesis of pyruvate synthesis of acetyl CoA enters into the Krebs cycle. The products enters into the electron transport chain. Then oxidative phosphorylation, synthesis of ATP molecule along with water and carbon dioxide. Okay, so this is what the overall reaction we have seen till now in the respiration. From the glucose, glycolysis, pyruvate, acetyl CoA, Krebs cycle, electron transport chain, oxidative phosphorylation, ATP synthesis, liberation of carbon dioxide and water. Okay, so we have seen in a particular sequence, but normally in a cell environment, in a in vitro, in vivo condition, normally the process they won't happen in a particular sequence. Okay, always we are not going to eat only the glucose. We all know that we are consuming proteins, fats and other organic substances for energy. Okay, so what will happen over here? While we are consuming fat or proteins, first they will enter into glucose or some other intermediary molecules such as pyruvate, then it will enter into the normal pathway as we have seen in the aerobic respiration. In other way, sometimes the body, the animal body or the plant system, they may require proteins and fat, and from the pyruvate they may enter into the synthetic mode. So the products of pyruvate they may enter into some other pathway or they may be utilized by some other pathway. So we cannot always predict the exact amount of energy is being produced from each and every glucose molecule, but we can predict. Okay, so we can assume, what if there will be no digression from the particular sequence of pathway, then what will be the amount of net ATP production from the particular aerobic respiration. Okay, you got my point? From the glucose till the production of the particular ATP and the products of carbon dioxide and water. Okay, if there is no digression, if there is no simultaneous synthesis or production, what will be the net amount of ATP will be produced in the particular sequence of aerobic respiration? That is what we are going to discuss now, the amount of ATP production in a aerobic respiration and while compared to the anaerobic respiration. And now we are going to discuss the net ATP production possibly from the aerobic respiration. As I told you, there are major events going to happen over here, the glycolysis, and from the glycolysis the synthesis of the pyruvate. And the next major event, as I told you, the interlinking in between the Krebs cycle and the glycolysis, that is the formation of acetyl CoA from the pyruvate, and then the Krebs cycle, and then finally the electron transport chain. As I told you, electron transport chain, they are going to receive the products of all these things and they are going to synthesize the particular ATP molecule. And first recall the ATP production from the glycolysis. As I told you, in the glycolysis, as we have seen in a glycolysis, from the glucose till the pyruvate, okay, there are two stages they are going to produce the particular ATP molecule. So there are two ATP molecules produced for each and every glucose molecule. Okay, so all these calculations for per glucose. Okay, so in glycolysis there are two ATP molecules being produced and there are two NADH molecules being released in the particular glycolysis. As we have seen in the glycolysis, we all know that from the glyceraldehyde 3-phosphate through the 1,3-bisphosphate, they are gonna release NADH molecule. Okay, so this is for single pyruvate or this is for single site. As I told you, there are two sites are going to happen. So glycerol 3-phosphate will be converted into dihydroxyacetone phosphate and they will be converted back to the particular glycerol 3-phosphate and they will release the NADH during the formation of 1,3-bisphosphate. But the point you have to remember over here, there are two NADH molecule is being synthesized before the pyruvate in the particular glycolysis. Okay, and till the pyruvate formation there are two ATP molecules and two NADH molecule we have produced in the glycolysis. And next, pyruvate to the acetyl CoA. the synthesis of acetyl-CoA, there will be no direct ATP production over here, and the enzyme dehydrogenase over here, they are going to liberate the NADH molecule. There are two NADH molecule again. For each and every pyruvate, there will be one NADH. For every glucose, there are two pyruvate molecules. So there are two NADH molecules being released per glucose. Okay, this is what you have to remember. Then enter into the Krebs cycle. Acetyl-CoA enters into the Krebs cycle, and over here, as I told you to remember that from the succinate to fumarate, they are going to synthesize. From the inorganic phosphate and the GDP, they are going to synthesize ATP, and as I told you, it is the equivalent of ATP. Okay, so GTP is equivalent to ATP, and by means of enzyme phosphokinase, they are going to create the molecule ATP from the GTP. Okay, so we have to consider this as ATP in the Krebs cycle. So for each and every pyruvate, there is a single ATP going to produce. For each and every glucose, there are two ATPs being produced. So you have to add this to the calculation. In the Krebs cycle, there are two direct ATP molecules. Actually, the produced molecules are two GTPs, and we are considering this as 2 ATP because GTPs are equivalent to the ATP, that is with the help of enzyme phosphokinase. Okay, so the point you have to remember, there are 2 ATPs in the from the Krebs cycle, and for each and every cycle there are 3 NADH molecules being produced. For each and every glucose there are 6, as I told you, 3 per pyruvate, 6 per glucose. This is what you have to always remember. You are not supposed to get confused with the number of molecules over here, because 2 pyruvate is being released from single glucose. This is what you have to always remember. So there are 6 NADH molecules being produced, and there are 2 FADH molecules. That means single FADH2 molecule released in a cycle of Krebs, and there are 2 FADH2 totally from per glucose molecule. Okay, and now we are going to calculate the total amount of ATP molecules from the particular glycolysis metabolism. As I told you, so these are all the direct ATP molecules. There are totally 4 ATPs. Okay, and from here, as I told you, these things, the NADH and FADH2 molecule, they are going to enter into the electron transport chain to synthesize the particular ATP molecule. And as I told you, one NADH molecule is equivalent to 3 ATP molecules. Okay, this is what you have to remember. One NADH molecule can involve in the synthesis of 3 ATP molecules. So this 2 NADH is equivalent to 2 into 3, 6 ATP in the electron transport chain. This is what you have to remember. So over here, the FADH2 is equivalent to 2 ATP molecule. Okay, so it is equivalent to 4 ATPs in the electron transport chain. Okay, and over here 6 into 3, 18 ATPs, and over here 2 into 3, 6 ATPs. Okay, so this is what the calculation we are gonna do. So this 6 ATPs from the 2 NADH produced in the glycolysis. These 6 ATPs produced from the conversion of acetyl-CoA from the NADH form, and these 18 ATPs from the NADH from the Krebs cycle, 4 ATPs from the FADH2 from the Krebs cycle. Okay, this is what you have to remember. Now we are going to add up all the things. 22, 28 and 34 ATPs from the electron transport chain. 4 ATPs we have already produced in the form of ATP. So totally it will be 38 ATPs. Okay, 22, 28 and 34. So this is just an approximate number, and you have to remember the ATPs is being utilized for some other steps in the glycolysis and other pathways. Okay, so this is just an approximate pathway, and you have to remember it will be around 34 to 36 normally. 36 ATPs we can produce from a single glucose metabolism. Okay, And these are all the things you have to remember about the respiration. So what and all we have seen in the respiration? We have started off with the what is the respiration and what are all the processes gonna involved in the respiration: aerobic respiration, anaerobic respiration, fermentation, and substrate level phosphorylation. Then we have seen something about the glycolysis, then the synthesis of pyruvate to the acetyl CoA entering into the Krebs cycle, then electron transport chain, oxidative phosphorylation, and then finally the calculations about the ATP production. Okay, and next thing you have to remember, the one more term you have to remember in the respiration, the amphipholic nature or amphipholic pathway. Okay, Krebs cycle is called the amphipholic pathway because there are two major metabolic pathways we are going to discuss: catabolism and anabolism. Okay, anabolism means synthesis of molecules, catabolism means breakdown of molecules. Okay, so normally we are calling this Krebs cycle as an amphipholic pathway because both the breakdown and the synthesis going to happen in a same pathway. Okay, so breakdown means they are going to break down the particular acetyl CoA, that is being synthesized from the pyruvate to some other molecule, and they are going to synthesize molecule, they are going to involve in the synthesis of the intermediates of the Krebs cycle. They are going to involve in the synthesis of amino acids and some other organic molecules. Okay, so that we are calling this Krebs cycle as an amphipholic pathway. Okay, and these are all the details you have to remember to answer the particular questions from the particular unit. Okay, and from the next class we are going to discuss the questions. What are all the questions we can expect from the respiration in higher plants.